Hey guys, this is the rest of the chapter for chapter 12. Hopefully everything's going okay for you. Based on your comments, it looks like for most of you it's going pretty well, which is good. You know, we just kind of have to hang in because it looks like we're going to be doing this for a while. So right now we're going to talk about DNA replication. So in the think about it slide here, it says before a cell divides, its DNA must be copied. How might the double helix structure of DNA make that possible? Now, you guys have done the gizmo, so you should have some idea of how this is going to work. So, what role does DNA polymerase play in copying the DNA? That's one of the questions here. So, DNA polymerase is an enzyme that joins individual nucleotides to produce a new strand of DNA. So base pairing in the double helix explained how DNA could be copied. So we have the A's and T's and G's and C's, and that's basically our code. Because each base on one strand pairs with only one base on the opposite strand. So because the A bonds with T and the C bonds with G, that means you can code the material. Each strand of the double helix has all the information needed to reconstruct the other half of the mechanism of base pairing. So if you split that right down the middle, both halves can now make a full copy by attaching new particles to make the other half. Because each strand can be used to make the other strand, the strands are said to be complementary. And we'll show you what we mean by that. Before a cell divides, so this is in the S phase of the cell cycle. That's one of the reasons why we went through that whole thing. If you remember, it goes G1, S, and then G2. And the S part of the cell cycle was DNA synthesis. And that's what this is. This is when this happens. The process ensures that each resulting cell has the same complete set of DNA molecules, which is very important. So here's the process. So during replication, the DNA molecule separates into two strands and then produces two new complementary strands following the rules of base pairing. Each strand of the double helix of DNA serves as a template or model for the new strand. So if this is getting unzipped here, so you can kind of see where you're basically unzipping this right down the middle of the chain, and then this DNA polymerase, which is an enzyme, goes along this and starts to add the opposite side. And at the same time, it's going to happen here as well. So you started off with one strand, you unzipped it right down the middle, and then you added the other side to the part that was unzipped. So if you could imagine taking a zipper, unzipping it, and then adding another zipper to each side, you would end up with two full zippers. And that's basically what's going on here. So how does DNA replication differ in prokaryotic cells and eukaryotic cells? Remember, eukaryotic cells are what we have. Prokaryotic cells are what bacteria have. Replication in most prokaryotic cells start from a single point and proceed in two directions until the entire chromosome is copied. How does DNA replication differ in prokaryotic and eukaryotic cells? Another way is that the replication begins at dozens or even hundreds of places on the DNA molecule in eukaryotic cells. So ours is much more complicated, proceeding in both directions until each chromosome is fully copied. It would just take way too long if we were to just start at one point and then just go forward. The cells of most prokaryotes have a single circular DNA molecule in the cytoplasm containing nearly all the cell's genetic information. Eukaryotic cells, on the other hand, have up to 1,000 times more DNA. Nearly all of the DNA of eukaryotic cells is found in the nucleus. And that's it for this. So let me just go back to this slide because this is really the big take home here today. Is you start off, you unzip the molecule right down the middle, and that's done with one enzyme. It's not super important that you know what that enzyme is called here, but Just know that there's an enzyme that goes through and unzips the DNA right in the middle, and then the DNA polymerase just adds the other side to it. So you end up with two copies of that are totally identical to what they started with.